Good morning to all. Welcome to the Diamond Group of Institutions, NEET and K-CET online classes. Today's class is biomolecules. That's some questions regarding as a previous questions from the NEET and the aims. So those are some of the two functional group characteristics of sugars are. So if you are taken as a sugar structures, so in these are the sugars, two functional groups are, we have to identify the two functional groups of the sugars are, they given as hydroxyl and methyl, carbonyl and methyl, carbonyl and hydroxyl, carbonyl and phosphate. See now, methyl groups are not present and phosphate groups are not present into the all sugars. Some of the sugars only they contain as a methyl group and phosphate groups. So if you are taken as a structure of the structure of the sugars and functional group of the sugars or common sugars are mostly they given as a when we seen as a sugars. So the sugars they contain as a carboxylic groups and hydroxyl groups. So common functional groups are the sugars are carboxylic group and the hydroxyl group. So here carboxylic groups are also called as carbonyl. Carboxylic groups are also called as a carbonyl and hydroxyl groups. So these are the functional group of the sugars that contain as a carbonyl or carboxylic group and hydroxyl groups. Answer is C. And the next question, a proteinaceous enzyme is. So here proteins are the polymer of amino acid. They are linked by the peptide bonds. A non-proteinaceous enzyme is. So all of the enzymes are not a proteins. So there some one exemption condition we are going to discuss. Some of the enzymes are they are not belongs into the protein. So here some are the given options. Lysozyme. Lysozyme is an enzyme. It is used to cleave in the or break down the cell wall of bacteria, and it is a proteinaceous enzyme. And ribozyme. Ribozyme. It is seen into the RNA molecule, that is ribosomes. Ribozyme it's not a proteinaceous enzyme. Ribozyme it's a nucleic acid. It's a nucleic acid. So ribozyme is an enzyme. It is seen into the prokaryotic organisms that contain as a 23S type of the ribosomes. That 23S type of the ribosomes they contain as a ribozyme enzyme, and it is non-proteinaceous. So exemption condition of the enzymes is all enzymes are not a proteins. Some of the enzymes are nucleic acids. That is ribozyme enzyme. And lydase. Lydase is the enzyme is used into the binding of the two DNA strands. It's also the proteinaceous enzyme. And deoxyribonuclease. So deoxyribonuclease enzymes also the proteinaceous enzyme. So out of the given four options, non-proteinaceous enzyme is ribozyme. Ribozyme it is a nucleic acid. It is seen into the RNA molecule. And the next question, which one of the following statement is wrong? So option A, uracil is a pyrimidine. So when we seen into the nitrogen bases, that nitrogen bases are N2 bases are classified into purines and pyrimidines. Purines are 2, adenine and guanine. Those are double ring structures. Pyrimidines are 3. Those are cytosine, thymine and uracil. These are the single ring structures. Instead of the thymine, uracil it was present into the RNA. So now here uracil is a pyrimidine. Yes, uracil is a pyrimidine. And option D, glycine is a sulfur containing amino acid. Glycine is a sulfur containing amino acid. Sulfur containing of the amino acids are 2 only. Those are methionine and cysteine. So these 2 of the amino acids only that contain as a sulfur in this structure of the amino acids of methionine and the cysteine. And here they given as a glycine is a sulfur containing of the amino acid. So glycine is not a sulfur containing of amino acid. And so glycine is a neutral amino acid. It's a neutral amino acid. Sucrose is a disaccharide. So when we seen as a carbohydrates, so carbohydrates are with all as a sugars, with all as a simple sugars, and these are the simple sugars are based upon the presence of sugar molecule. We are classified into monosaccharides, disaccharides, and polysaccharides. So disaccharides means when we done the hydrolysis of the sugars, they can form into the simple sugars. So, so these are the sucrose is a disaccharide. Disaccharides are that undergoing as a hydrolysis, and they can form into two simple sugars. So such type of the sugars are we call as a disaccharides. So sucrose is a disaccharide only. And option D, cellulose is a polysaccharide. Cellulose is a polysaccharide because when we done the hydrolysis of cellulose, many number of polymer of glucose molecules will be going to form. So that's why this cellulose is a polysaccharide molecule. So question they are asking, which one of the following statements is wrong? Out of the given options, A, C, D are correct statement only, B statement only wrong. So answer is B. And next question, the chitinous exoskeleton of arthropods is formed by the polymerization of. So arthropods are most abundant component, second most abundant element or component in the living organisms are the living organisms are that contain as a living organisms are they contain as a arthropods, and these are the arthropods are exoskeleton bodies are made up of by. So here they are given as option N-acetyl glucosamine, lipoglycans, and keratinosulfate, and chondrian sulfate, and D-glucosamine. So here the exoskeleton bodies of arthropods are made up of by N-acetyl glucosamine. It is a polysaccharide. N-acetyl glucosamine. So in these are the N-acetyl glucosamine. It's a certain abundant component in a living organisms. Certain abundant component in a all living organisms. These are the N-acetyl glucosamine. We are also called as NAD. That N-acetyl glucosamine is a certain abundant component in the all living organisms, that is into the arthropods. The arthropods exoskeleton bodies, that is chitin exoskeleton bodies is made up of by N-acetyl glucosamine. So option answer it is a N-acetyl glucosamine, and remaining as a lipoglycans and D-glucosamine. These are the D-glucosamine is a structure of the glucose, and lipoglycans are we are seeing into the plasma membranes, and keratin sulfates and the chondrian sulfates are used to the preparation of the shampoos. So answer is for the exoskeleton of arthropods are made up of by the N-acetyl glucosamine. Next question, which of the biomolecules does have a phosphodiester bond? See here, phosphodiester bonds are their presence in between the two successive nucleotides. So when we seen about that phosphodiester bonds, generally that nucleic acids are made up of by the polymer of nucleotides. In between the nucleotides, from the one nucleotide into the other one of the nucleotide, a phosphodiester bond will be going to present. So imagine this is the one nucleotide and other one of the nucleotide. In between the two nucleotides, two successive nucleotides are connected together by the presence of phosphodiester bonds. So now, which of the biomolecules does have a phosphodiester bond linkage? So now options are A, amino acids in a polypeptide. So amino acids are connected together in a polypeptide chain by the peptide bond. Peptide bond it was present in a polypeptide chain. Nucleic acids in a nucleotides. So nucleic acids are the polymer of nucleotides. These all of the nucleotides are. Nucleotides depend as a nitrogen base, sugar and phosphate molecules are combined together which all as a nucleotides. So here this is the one nucleotide and this is the other one of the nucleotides. These nucleotides are connected together by the presence of phosphodiester bond. This is the one ester bond and this is the other one of the ester bond connected together with the phosphate molecule. So that's why it's called as a phosphodiester bond. The phosphodiester linkage it is seen into the nucleic acids of nucleotides. And option C, fatty acids of diglycerides, monosaccharides of a polysaccharides. So here monosaccharides in a polysaccharide, those are made up of by they connected together by glycosidic linkage. Glycosidic linkage. And fatty acids in a diglycerides. Fatty acids in a diglycerides also they are connected together by the glycosidic linkage. So out of the four options, phosphodiester bond we are seeing in a nucleic acids in a nucleotide. That polymer of nucleotides are they connected together. And next question, select the option which is not correct with respect to enzyme action. Select the option which is not correct with the enzyme action. Option A, substrate binds with the enzyme and at its active site. So you know the enzyme actions. So substrates are they bind into the active site of the enzyme. At the time of the enzyme action, we are seeing some of the steps involved in enzyme action. You can imagine about this is the enzyme and this is the active site of the enzyme. So these are you can imagine substrate. So that substrates are they can bind into the active site of the enzyme. That is the first step of the enzyme action. And option B, addition of a lot of succinate does not reverse inhibition of succinic acid dehydrogenase by malonate. So here actually that addition of succinate does not reverse the inhibition of succinic acid dehydrogenase by malonate. So these are the succinic acid dehydrogenase enzyme. It will be inhibited by the presence of malonate. So this is called as a competitive inhibition. Competitive inhibition. Inhibition it was classified into two types, competitive and non-competitive. In the competitive inhibition, it is in between the that is inhibitor and substrate. So to the binding of the active site of the enzyme, in between the inhibitor and substrate competition will be present. Such type of the inhibition we call as a competitive inhibition. So for this are the competitive inhibition, better example as a that is prevention that inactivation of the succinic acid dehydrogenase enzyme by the malonate. So that's why this is called as a competitive inhibition. But they given as here by adding of the much concentration of succinate, the succinate dehydrogenase enzyme is not inhibited. So does not reverse the inhibition of succinic acid dehydrogenase enzyme by the malonate they given, but the succinic acid dehydrogenase enzyme is inhibited by the presence of malonate. So in that statement may be. And option C, in a non-competitive inhibitor binds the enzyme, in a non-competitive inhibition binds the enzyme at a active site of distinct from the which binds to substrate. So in the competitive inhibition, competition presents in between the inhibitor and substrate molecules. But in a non-competitive inhibition, in these are the non-competitive inhibitions, these are the inhibition and substrates will be done to bind and they can form into the, these are the inhibition and substrate will be done to combine together, and later these are the substrates will be done to bind into the active site of the enzyme and they can form into enzyme substrate complex. So now in the non-competitive inhibitor binds the enzyme at a site distinct from the which binds into substrates. So these are the substrates are they bind into the enzyme molecules. Those are different. So and option D, malonate is a competitive inhibitor of the succinic acid dehydrogenase enzyme. So here only we are discussed B and D options. In the B option they given as a reverse and D is separate. Malonate is a competitive inhibition of the succinic acid dehydrogenase enzyme. Option D is the correct statement. And option C, in a non-competitive inhibition, substrates are they bind into the active site of the enzyme. C also is the correct. And the option A, substrate binds with the active site of the enzyme. This is the first step of the enzyme action. Option A also correct. In the correct statement it is B only. Answer is B. In the next question, Which of the following is a non-reducing carbohydrate? So which of the following is a non-reducing carbohydrate? So when we can something as a non-reducing carbohydrates. Carbohydrates are, which of the carbohydrates are, they won't have a free aldehyde and the ketone groups. Such type of the carbohydrates are we call as a non-reducing carbohydrates. So if you are taken as examples in this, maltase, maltose, sucrose, lactose, and ribose 5 phosphates. Out of these four, maltose, lactose, and ribose 5 phosphates are they contain as a free aldehyde groups or carboxylic groups or ketonic groups. But sucrose molecule only does not having as a free aldehyde and ketonic group. So that's why non-reducing carbohydrate is a sucrose only. Answer is B. Next question. Transition state structure of substrate formed during the enzymatic reaction is. So now, at the time of conversion of substrates into a product, enzyme and substrates are then involved. So these are the enzyme and substrates are that combined together that can form as the intermediate state that is called as enzyme substrate complex. And later these are the enzyme substrate complex will be going to become as a enzyme product. So here enzymes plus the substrates we are going to add it, and these enzyme substrates are combined together that can form as the intermediate or unstable component we call as a enzyme substrate complex. These are the enzyme substrate complex later converted into the enzyme product. So now here at the time of the conversion of the substrates into the products, so these are the intermediate stage will be going to form at the action of these enzymes. That intermediate stage it is also called as a transient stage. So in these are the transient stage is a unstable. So the question asking as transition state structure of the substrate formed during an enzymatic reaction. So they given as a transient and unstable, permanent and stable, transient and stable, permanent and unstable. Out of the given options at the time of the enzyme actions, conversion of the enzymes into the enzyme substrates into the enzyme products, intermediate stage is formed, that is only we call as enzyme substrate complex. That enzyme substrate complex is a transient and unstable. It is an unstable component. After the formation of the enzyme substrate complex, immediately they converted into the enzyme product. So that's why transient, unstable is a right answer. Answer is A. And the next question, which of the following statements about enzyme is wrong? Which of the following statements about enzyme is wrong? Option A, enzymes are denatured at a high temperature. Yes, enzymes are denatured at the high temperature. Normally, enzymes are there will be denatured after the 40 degrees temperature. Some of the enzymes are there having exemptions. Those are the enzymes they are involved in a PCR technique, polymerization chain reaction. So these are the enzymes that can survive even though the high degrees temperature. These are the enzymes they will be drawn to active even though the high degrees temperature of 80. So such type of the enzymes will be collected from the specialized bacteria, that is thermophilic bacteria. So enzymes are denatured at a high temperature. If the temperature is increased, activity of the enzyme will be done to stops. That's called as denature of the enzymes at the high temperature. And enzymes mostly proteins, but some are lipids also. Most of the enzymes are, all of the enzymes are proteins, but some of the enzymes are not lipids. Some of the enzymes are nucleic acids. Some of the enzymes are nucleic acid. That nucleic acid enzyme it is ribozyme. And option C, enzymes are highly specific. Yes, enzymes are highly specific And option D, enzymes are require optimum pH and temperature for maximum activity. The activity of the enzyme is depend upon the temperature and also the pH value. See, if the pH value and the temperature in the environment, pH value and temperature, any changes that takes place means automatically activity of the enzyme will be done to change, or activity of the enzyme will be done to prevent. So enzymes are highly specific. Specific enzymes that can work into the specific functions. So other enzymes are different enzymes they will not perform into the other functions. So specific enzymes they can work into the specific functions only. So out of the given four options, incorrect statement about the enzyme it is B. So they given as enzymes are mostly proteins, but some are lipids. So these are the some are not a lipid, some are the nucleic acid. That nucleic acid enzyme it is ribozyme. Answer B. And the next question, uridine is present in only RNA is. Uridine present only in RNA is. So here uridine is a component, it is present in RNA molecule. This uridine is a option A, nucleoside, nucleotide, purine and pyrimidine. So first you want to know uridine, it is belongs into the pyramid, that is uridine, it belongs into the nitrogen base. These are the nitrogen bases are classified into two types, purines and pyrimidines. So here the pyrimidines are thymine, cytosine and uracil. Instead of the thymine, uracil it was present, but here not given as a uracil. If they given as a uracil, we can consider as a pyrimidine. So here not given as a uracil, it's a uridine. Uridine and the uracil differences it is. Uracil is the only nitrogen base, and these are the nitrogen bases are combined into the sugar molecules. That is becoming as a uridine. So uridine nitrogen base are the uracil and the sugar molecules, that is called as uridine. That uridine molecule, sugar and nitrogen base are combined together, we call as a nucleoside. Nucleoside. So sugar, nitrogen base and the sugar and nitrogen bases are in between the nitrogen base and sugar and glycoside bond will be not present. That is called as a nucleoside. Answer is a nucleoside. So why not as a pyrimidine? Pyrimidine means it's a uracil only. Only nitrogen base should be present. But here they given as a uridine. It is associated into the sugar molecule. And why not considering as a purine? Purines are two adenine and guanine. So purine is not right. And the nucleotide, why we are not considering as a nucleotide? If they given as if that uridine is considering as a nucleotide. Nucleotide means nitrogen base, sugar and the phosphate molecules should be present. If the phosphate molecules are present means uridic acid will be don't form. That acid name was not given, so that's why it's just given as a uridine molecules. Nitrogen base of the uracil is associated into the sugar molecule that can form as a uridine is a nucleoside. Answer is A. And the next question, cellulose is a major component of cell wall. Cellulose is a major component of cell wall of. Options are given as a Pseudomonas, Saccharomyces, Pythium and Xanthomonas. So see now, cellulose is a polysaccharide. These cellulose molecules are they connected together by the glycoside bond, and these cellulose molecules are we are seeing a cell wall of plant. So now these are the cellulose is a option component cell wall of. Option A, Pseudomonas they given. Pseudomonas is a bacteria. Bacterial cell wall is made up of by NAM and NAD. NAM and NAD. N-acetyl muramic acid and N-acetyl glucosamine. So bacterial cell wall it's not contained as a cellulose. And Saccharomyces, it's a fungi. So that fungi cell wall is made up of by chitin. And Pythium. Pythium also is a fungi only. One of the fungi excluding the cell wall, it's not contained as a chitin. It's made up of by cellulose. So one of the fungi, its cell wall is made up of by the cellulose. That fungi name it is Pythium. The Pythium hyphae cell wall is thin than the other fungi. And Xanthomonas. Here Xanthomonas is a bacteria. Bacterial cell wall is made up of by NAM and NAD. So cellulose is a major component cell wall of Pythium. So this Pythium is a fungi, and one of the fungi it is instead of the cell wall chitin, here these are the cell wall of the Pythium, it is made up of by cellulose. So answer is C. And next question, which of the following nucleotide sequence contain 4 pyrimidine bases? So again, question from the nitrogen bases only. Pyrimidines are belongs into cytosine, thymine, and uracil. So out of the given sequence of nucleotide, which are the nucleotide sequence that contain as a 4 pyrimidine bases only? So now you see the option here. G is a purine, adenine is a purine, thymine is a pyrimidine, cytosine is a pyrimidine, and adenine is a purine, thymine is a pyrimidine, and guanine is a purine, and cytosine is a pyrimidine. So by a pyrimidine C, T, and TC. Here the 4 nitrogen bases in the first option only. 4 nitrogen bases are then contained as a pyrimidines. See now the second option also, if you will get any doubt here. So guanine is a purine, cytosine is a pyrimidine, uracil is a pyrimidine, and remaining they given as a adenine purine, guanine purine, adenine purine, and cytosine is the pyrimidine, and remain as a purine and purine. So here 3 only given. So out of the 4 options, answer A, it's contained as a thymine cytosine and thymine cytosine, 4 nitrogen bases as a pyrimidines. Remaining options are in the option B, they contain as a 3 pyrimidines, and option C, they contain as a 3 pyrimidines, and so here option 2 they contain as 3 pyrimidines, and option C also they contain as 3 pyrimidines, but option 4 only they contain as a 4 pyrimidines. So answer for the option A only they contain as a 4 pyrimidines. Answer is A. So those are TC and TC, these are the pyrimidines. Next question, which of the following groups are all polysaccharides? So here polysaccharides, poly means many, saccharides as a sugars. Which are the given option, it's contain only the polysaccharides. So options are sucrose, glucose, and fructose. See now, sucrose, glucose, and fructose. Sucrose is a disaccharide, glucose is a monosaccharide, and fructose also the monosaccharide. Maltose, lactose, and fructose. Maltose is a disaccharide, lactose is a disaccharide, fructose is a monosaccharide. Glycogen, sucrose, and maltose. Glycogen is a polysaccharide, sucrose is a disaccharide, and maltose is a disaccharide. And glycogen is a polysaccharide, cellulose also polysaccharide, and starch also the polysaccharide. So in these all of the option D, glycogen, cellulose, and starch, those all are made up of by the polymer of starch, polymer of glucose, and these are the glucose molecules are they connected together by the glycoside bond. 1 term of 4 glycoside bond, they are connected together. So option D, glycogen, cellulose, and starch, they contain as a polysaccharide. So answer is a D. And next question, which is wrong about nucleic acids? So DNA is a single stranded in some viruses. Yes, DNA is a single stranded in some viruses. It's a third statement only. RNA is a double stranded in occasionally. So some of the organisms, RNA also is present in double stranded. TMV example, and for these 5174 bacteriophage example for the double stranded DNA. Sorry, for the single stranded DNA. And length of helix is 45 angstroms in BDNA. BDNA is a balanced DNA. It is a stable DNA. So this BDNA, the length of the helix we are also referred as a turn or coil. So that length of the helix or turn of the helix or coil of the helix in a BDNA, it is 34 angstrom units, otherwise 3.4 nanometers. It's not a 45 angstrom units. And option D, 1 turn of zDNA has 12 base pairs. So zDNA is a type of DNA. It's also called as the zigzag DNA. These are the zDNA. It's contained as a each turn or each coil or each helix. It's contained as a 12 base pairs. So out of the given options, DNA is a single stranded in some viruses, in example as 5'74". RNA is a double stranded, occasionally it is an example for TMV. And length of the helix is 45 angstrom units in bDNA. It is a 34 angstrom units. It should be. So entire statement is C. And option D, one turn of the ZDNA has 12 base pairs. Yes, ZDNA, zigzag DNA contain as a 12 base pairs per each helix. Answer is C. The next question, which is not considered with double helical structure of DNA? Which is not consist with the double helical structure of DNA? Option A, adenine equals to thymine, cytosine equals to guanine. So when we can see the apply the Chargaff rule, number of purines are equals into the number of pyrimidines. Always adenine pairs with the thymine, cytosine pairs with the guanine. Number of thymines are equals into the number of adenine, and number of guanines are equals into the number of cytosine. So it's a correct statement only. And option B, density of DNA decreases on heating. Density means weight of the DNA will be don't decrease by the heating. If the DNA will be don't heat, so the two strands of the DNA they are connected together with the hydrogen bonds. So if you are doing the heating of the DNA, the breakdown of the hydrogen bonds will don't take place. Double strand DNA is converted into a single stranded. Density will be going to decrease. So option B also correct. And option C, A by T plus G by A by T by G plus C is not constant. So A by T and G by C, sorry, A by T, A plus T by G plus C is a constant. So they given as a not constant. Incorrect statement of this DNA, it is C only. Answer is